induce ovarian insufficiency in the mice, can we restore ovarian function? That was really the crux of the work. So how to test that? So there were two sets of workflows. So this is the way these are generated in the lab. So collect granulosa cells, in this case from mice that have been stimulated, transduce them using them in knockdown factors to get these murine grips, granulosa cell-induced pluripotent stem cells. In order to be able to trace the lineage of the cells, we infected them with GFP, right? So that you knew that the green cells are the ones you have reprogrammed. And then take them through this embryoid body differentiation. This, again, the spontaneous process that will happen. So you let them form embryo bodies, you put them on gelatin-coated plates, they'll attach, and then use flow cytometry to sort out the ones which express the AMH receptor. So you want cells that are both GFP positive and express the AMH receptor, and then look at their steroidogenic capacity as well as their germ cell marker capacity. So that was the first set of experiments. And essentially, if you compare a normal mouse ovary versus what we see in these sorted embryoid bodies, we see the same markers that we see in a mature ovary. We will see in the oocytes, in these embryoid bodies that are derived from these granulosa cells. So we saw oocyte markers. The other thing which was interesting in this particular study, this sort of came out in the course of developmental work, was it turns out if you keep some of the follicular fluid in the culture, this works a lot better. So there's something about, it's not that different than using conditioned media, but if you take the follicular fluid and you add either 1% or 5% follicular fluid back into the culture, it dramatically increases the efficiency of the polarization. So you get
mouse follicular fluid.
Mouse follicular fluid. So human follicular fluid. Sorry, it's human follicular fluid added to mouse culture.
Okay.
Sorry. So whether it's human or mouse, you can do this with human and mouse cells, but it's human follicular fluid. You get liters of human follicular fluid from IVF. So, so you get higher production of estradiol, as well as higher expression of the germ cell markers. So it's not just, okay, you're putting more substrate in and so they're making more estradiol. You could sort of imagine that. But actually you get a higher expression of these primordial germ cell markers. Now, this process, so then that was the in vitro part. So what about the in vivo side of it? So again, sorting out these cells, it's a very small population of cells, we're talking about 0.7% of a million cells. But if you take the unsorted cells, so you have the ones that you haven't picked out the AMH receptor positive, and you inject them into a mouse, they'll form a teratoma, right? That's how we showed the COVID. But if you take the sorted ones, they don't form a teratoma. Now, we didn't see anything. What was interesting is if you take the mice and you look at their hormone levels, they're making estrogen. So there's no teratoma, but you correct the endocrine defect in the mice. Okay.
Is this like you're just delivering the cells IP or something?
In this case, we were injecting them. Well, we do inject them into the ovary of the mice.
Yeah.
But I was able to figure out which mice, just by looking at them, had received sorted cells or didn't, because, well, just on the tumor, when you give the mice chemo, we'll talk about in a second, the ones which end up with ovarian insufficiency, their mammary glands go away. Whereas I could tell you which mouse had received stem cells, which ones didn't, because I could see the mammary development of the mice. So just picking them up, you can tell which ones are the controls.
So teratoma, was it just look like a benign teratoma? Or?
Right, well, yeah, exactly, yeah. Because that's usually the way you would test for potency. But in this case, we actually don't see it this way it says. Okay, so this is the crux of the in vivo experiment, was to take 8-week-old nude mice, gave them alkylating chemotherapy with cyclophosphamide busulfan, which is one dose is highly effective at inducing endocrine insufficiency in the mice. Then we would perform laparotomies in the mice, and into the ovary inject either just vehicle, the sorted cells, or the unsorted cells. Okay. So the unsorted ones would form these huge teratomas in the mice, and the mice would actually eventually die because they get these huge tumors. the control mice, we would see that they developed ovarian insufficiency. They made no detectable levels of estradiol. Sorted mice, though, well, we'll talk about that. So, as I said, we GFP labeled them so we could tease out where the cells ended up in the ovary. And this is what we found. So if we injected these sorted induced pluripotent stem cells into the ovary, we could see green oocytes. This is what I call the green eggs in mouse experiment. Right? So, very happy day in the lab to see a green egg come out of an ovary of a mouse. So we said we got GFP positive oocytes from ovaries which had been ablated with chemotherapy. Then we took those oocytes and were able to both activate them through parthenogenesis, but also to fertilize them with sperm in the lab to form zygotes. What was also interesting is that if you looked at the ovaries of the mice, we found GFP negative oocytes as well, surrounded by GFP positive granulosa cells. Meaning that what had happened was that some of the stem cells, when we put them into the mice, had gone down the steroidogenic differentiation pattern, but actually were now surrounding oocytes and seemingly rescuing them. So then the question was, okay, is it possible that granulosa cell loss is an unrecognized cause of chemotherapy-induced infertility? So maybe what happens is you kill off the granulosa cells, and so the oocyte dies. It's not that you kill the oocyte. But if you think about it, an oocyte develops in utero, right? So it lives for 50 years before it dies. The thought that a dose of chemotherapy is suddenly going to kill it, it's a pretty quiescent cell. It really should be quite tough. Granulosa cells, though, are not. And so it may be that the defect here is depleting the granulosa cells. And so it could be that if you repopulate the granulosa cell population of the ovary, that may actually rescue a lot of the oocytes. So this is, again, just looking at the steroidogenic, just from the blood of the mice. So if we look at the mice that got chemo, their estradiol levels fall precipitously, whereas we see that the mice that got the sorted cells, their estradiol levels go up. Their testosterone and progesterone levels don't go up. So these are estrogen producing cells. And then this is where things got really fun is, okay, you have neomutagenesis. So then we looked at, since these were injected into the ovary, we questioned whether or not the mice could spontaneously conceive. And they could, just spontaneously breeding. So we had day 18 embryos. We saw green embryos. When we took out ovaries from the mice, we could see green eggs in situ, and then they would have live pups. Now,
How do you know that this is neomutagenesis and not just retained primordial?
so we had to do maternity tests. So what we found actually was if you just look at the GFP, some of them are GFP positive and some of them are GFP negative. Some of them are retained oocytes, some of them are derived from the ones you injected. And we only injected one ovary.
Okay.
So what we would see is the other ovary, there were paraffin effects. You actually had retention of oocytes in the ovary that we didn't inject, as well as the one that we did. And we did, for each mouse, we did lineage tracing using SCR testing, just like you're on CSI, to figure out which pups were derived from the stem cells and which ones were native, right? Because the stem cell derived mice were a different mother, like a donor egg, essentially. And so this is showing several different litters of these mice. So the open arrows are GFP negative pups, and all the other ones are GFP positive pups, which is fun. So we would see these litters of, and they would be mixed. Some of them were oocyte derived, and some of them weren't. And phenotypically, they looked identical. Now then we questioned, well, what would happen to these pups? So we bred them to the F1 generation and the F2 generation. We found that the pups were fertile, and their offspring also were fertile and seemed to be normal. So they had normal litter sizes and seemed to have normal fertility. So the take-home idea here is, could this be an autologous stem cell-based model for fertility endocrine restoration? And that can you take granulosa cells, ideally autologously, although I can't think of any reason actually why you couldn't have granulosa cell donation. Generate them into iPSCs, and now you would have a stem cell that, when injected into the ovary, and I think this is, we were talking about John's work before, this is I think one of the key things, it's putting these cells into the ovarian stroma is quite key to get them to differentiate in a native way into both oocytes as well as granulosa cells. You could either create a system where you ex vivo are generating bioidentical hormones, or are you actually repopulating the ovary itself with a cell-based therapy?
You would be, if you use donor granulosa cells, you would be populating with donor genetics, right?
Well, maybe not, because it'd be probably a chimera.
I mean, if you get the granulosa cells salvaging them.
Correct. So because you could sort them for the ones that are steroidogenic, but not won't make germ cells. Yeah, so you could do that too. So just to wrap up, we know that chemotherapy-induced ovarian insufficiency is a large and growing public health burden. Granulosa cells are really a promising source to induce pluripotent stem cells that I would argue are uniquely positioned for this problem, much more than the idea of making iPSCs for fibroblasts or stem cells or things like that. And what's quite fascinating about granulosa cells is not only are you recapitulating the endocrine functions, you're recapitulating the reproductive functions of the ovary as well. And maybe this is a way to rescue oocytes from atresia. So obviously I want to thank Ray. This was really Ray's insight and genius which led to this, and the people in Ray's lab over the years who did mention his work, people from my own laboratory. My laboratory tended to handle the chemo and the animal work, and then other supporters from the RAI division, the great most supporting Mark Hornstein and Liz Ginsburg, who were great friends and mentors and advocates for this work. Thank you.
Questions? David?
Yeah, thanks. That was a great topic. In terms of the breadth too, in terms of the magnitude of the problem. Granulosa cells, there's a big difference between a mural granulosa cell and a cumulus granulosa cell. And, I really like the logic of let's try to go back to a not so differentiated state in terms of generating those lines initially. I'm not sure how many people are aware of it, but back in the days of somatic cell nuclear transfer, i.e. cloning, the most efficient cell to reprogram is a cumulus granulosa cell. Not only that, cumulus granulosa, and this work, Jonathan Verclone showed this in human cumulus cells, they have totipotency. So under different conditions, they will form adipocytes, they will form smooth muscle, they can go pretty much down all three embryonic germ layers. So in your experiments, you showed us some green eggs and you showed us some green granulosa cells. I would have thought you would also see green vasculature, green interstitial tissue, and other things. Was there a little more expression of, say, different cell types, different somatic cell types?
So not if you sort them based on the AMH receptor. That's the key. So you're absolutely right. If you took unsorted stem cells, for them to drive granulosa cells, absolutely, you'll see adipocytes, you'll see vascular cells, you'll see probably see all different differentiation. But if you allow them to do this little bit of spontaneous differentiation in vitro, and then you sort them based on those receptors, now they won't do that. Now they will only go down these ovarian differentiation pathway. And for whatever reason, and I can't explain exactly what it is in the follicular fluid, if you spike a little follicular fluid in there, it sort of nudges them in the right direction. Which is actually a technique that we've adapted in my own lab for other stem cell experiments, right? If you want to make a cardiac cell, well, take some cultured media from cardiac myocytes and spike that in your culture and make it stem cells move that way. So it's there could be something that is universal in that.
The mice that you were working with were all had induced ovarian insufficiency because of chemotherapy, because that's what you were interested in.
Right.
Did Ray ever try the sun senescent mice?
We never tried on senescent mice. What I will say about nude mice, though, is that nude mice do. So we did do it in nude mice just without chemo, and it works there as well because they do have accelerated follicular atresia. Mechanism that's not quite clear what that is, but it is a function of nude mice.
I was interested when you kind of mentioned that perhaps the oocytes are not aging, but the granulosa cells are aging because that goes in line with what we've been kind of hypothesizing
Yeah,
for about 15 years. Yeah.
Some other data on that. There has been some data that the oocyte itself really doesn't get, I think you mentioned that, damaged by chemo. It is, once it's getting recruited and starts its journey through the microenvironment of the ovary, that it gets damaged.
So, our hypothesis been if we could do something to improve the ovarian environment, we could possibly make better eggs.
Right. So I would say from a prep, obviously there's a lot of concerns about fertilizing induced pluripotent stem cells to make humans.
Sure.
But it could be that that's not the process you actually need. It could be that really what you need to do is supplement an ovary with granulosa cells, and the ovary may repair itself.
What do you think about the mitochondria? I mean, the nuclear transfer thing that we're trying to take the good mitochondria cytoplasm and then transfer it to another thing.
Perhaps we'll propose that, right? Can you take younger mitochondria and put them in?
Yeah.
Hasn't worked very well.
It may not be that that's really what the problem is.
Once
It's not the oocyte, it's the support cells around the oocyte.
Yes. Yes.
Once you resuscitate the ovary somehow, it's going to make its mitochondria.
That's true. Right. So it may not really be that the issue is aged oocytes at all. It might just be the fact that there's a natural granulosa cell decline. Senescent granulosa cells. Exactly right.
It's all in the microenvironment. I mean, in cancer, it's not different. I mean, that's now being recognized more and more importantly that it's the microenvironment that is really determining the fate of what is happening. So if we can get rid of the fibrosis, for example, and get some new granulosa cells in there.
Well, I think that's been one of the ideas, just coming back to the beginning of your talk. You said this is work that came out of Carla Hutt's lab. You mentioned Amy Winship.
Right.
Whether it's chemo or even they've done some work with radiation exposed ovaries as well. And there is a baseline level of DNA repair going on in even primordial oocytes. Okay. And of course, you boost it, but it's, there's much more damage in the granulosa cells, especially of antral follicles, okay, which are in a more proliferative state. So you can kind of expect that their cell cycle's moving so fast, they're going to be more susceptible to it. And that's kind of where the burnout hypothesis came from, which, by the way, is what Joramiro was suggesting, that early follicle pool, which is making the AMH, is what was suppressing primordial follicle activation.
Right.
So I don't know where that story has gone, but is there a third kind of model for the reason for the POI?
Well, that's a part of what I'm proposing, because I think there may be a granulosa cell centric.
Granulosa, or maybe some kind of ovarian stem cell that you need to kind of keep the niche going.
Right. And that the idea would be if you could maintain that microenvironment, then the oocytes would be fine. In other words, perhaps what you need to do, so again, there's a window of opportunity. So again, most of these patients are not going to have POI immediately after completing chemotherapy. As I said, it's usually a year, 2 years, 3 years lag. So what I've proposed is to say, well, no, what you really want to do is when someone completes chemotherapy, you know that she's at a high risk of developing POI. You infuse the granulosa cells into the ovary at that time, before the POI is fully developed, and then you'll prevent it from developing. In other words, it's it's a restoration or repair process. So if you intervene soon enough, then you won't get the follicular atresia.
Stay ahead of the game.
With
Right,
cancer,
yeah.
cancer patients, you have the opportunity to harvest granulosa cells before they get their chemo. I mean,
Even for the chemo, but even if you did it after they got their chemo,
Yeah.
right? So in other words, because you probably have a year or two
you
window
have
there.
that honeymoon time.
Right, where you can rescue those oocytes. So I would argue those oocytes haven't been damaged by the chemo. The follicular atresia is happening
That has
from
been
this
shown.
process.
That has been shown.
It's fascinating that it's the AMHR2 cells.
Yeah,
that are-
exactly.
Yeah,
Mhm.
right.
What is that all about?
Yeah, well.
That's the big question
Yeah.
since I discovered the story with all those receptors in the adrenal.
Yeah.
Why the hell are they there?
Right.
You know?
So are they similar origins? I mean, embryo.
Yeah, they have
embryonic.
adrenal and ovaries have a common primordial.
Yeah.
in the embryo.
Yeah.
So, and there have been experiments where people have induced follicle production in adrenals.
You can get germ cells in adrenal glands, for sure.
In
Yeah, I mean, germ cell
Yeah,
tumors.
yeah,
and
right.
adrenal glands.
Yeah. So it's, they basically come from the same stem cell. So there's still a lot to be learned, but it's also not surprising that the oocyte is not damaged, because when you think about it, how primitive the primordial follicle is, it has basically no supply. It's incomplete. It exists for years and years in complete isolation, more or less, with minimal energy need and nothing going on, until it gets activated.
Well, and one of the other things that makes the idea of sort of granulosa cell donation and granulosa cell transplant sort of interesting. We're talking about autologous granulosa cells, but the ovary being an immune privileged site, you could imagine that if you inject granulosa cells, even from another individual, it might not be rejected.
Yeah.
Yeah.
At least not very easily.
Yeah.
So I again, there's a good argument to say that it might be taken up very well by the ovary. That experimental, but at least in the now these mice are immunosuppressed mice, but even in mice that aren't immunosuppressed, they will take them, unrelated donors.
Was there any particular reason you went with the nude mice? I mean, a lot of the other work in this field has been with like Black 6 and,
So the donor mice are Black 6, right? But we used nude mice for two reasons. One, we really didn't know if they would reject the cells or not. That was one question. But also because we knew they would be easier to induce follicular atresia because they have this accelerated follicular atresia anyway, they're predisposed to it. So the argument that we made was that this is a better model of, say, BRCA1 carriers, right? Sort of a similar problem, and accelerated follicular atresia.
or outbreak, which probably,
Exactly right.
you might not have seen that result in an outbreak.
That's true.
Let me ask a clinical question to all the brains here. Can we, based on how much AMH is being done on everybody who shows up, I would say the 20 patients, 10 ask for an AMH because they're trying to freeze eggs. Can we predict who is going to have follicular atresia based on the level of AMH?
I'm not the one to ask.
Predict when and where.
When they're going to lose. I mean, many of them, the AMH is low and they're 30.
It hasn't proven to be very.
In my
cannot
experience, it hasn't, but
You cannot predict menopause based on AMH very reliably. I mean, it's a general trend, obviously, but you cannot predict it really with sufficient accuracy. But
The image that you're measuring is just a reflection of how many antral follicles
exactly.
are there. And-
I'll tell
and
you.
the other things will influence how many antral follicles will show up at a particular time. So it's not a constant indicator. Over time, obviously, people become menopausal, they have no AMH, but it's not a good way of telling the future.
I will tell you, most gynecologists right now offer AMH or test AMH, or not everybody,
And it's an indirect measure of
And-
somebody's potential of
it is
responding today.
and they
But-
send her to me to figure out what that's going on. And my typical lecture is, we cannot predict this, we can't tell this, we can't tell that. But does anybody know if there is any literature indicating that this woman is going to go into menopause in 5 years and will not going to have eggs?
Yeah, there's a lot of literature,
Yeah.
but it shows what was said here, that it is not a real good predictor, even though recently I saw a paper somewhere where they actually built a better model in some ways in predicting the age of menopause. But in principle, it is not a great model. At the same time, AMH, especially in older women, is clinically, I think, the best predictor. I mean, that, in an older woman, I take the AMH value any day ahead of the FSH. In younger women, I take the FSH as a predictor for their current status. Neither tells me when this woman will go into menopause, because there are lots of reasons why women can go into menopause.
These women are trying to plan egg freezing. That's why I say, that's why there's being screened.
But for egg freezing, again, it depends on the age, but if they're young, I think the FSH is to a degree more important than the AMH.
If you have a 23-year-old who has an AMH of 1.2, yeah, probably, she wants to freeze some eggs.
Yeah. Of course.
0.5 is what I'm saying.
Yeah, that's a problem.
Yeah.
She probably won't make it to 50.
I don't think she'll make it
That's
to
for
40.
sure.
But you can't predict when she's going to
Right.
come in.
Yeah, but the other side of the coin is that even if you take young women who are diagnosed with premature ovarian aging based on low AMH and or high FSH levels, that fact by itself does not predict that they necessarily will have early menopause. They may still have menopause at age 51.
And the other thing is, you're dealing with young people who might be using contraception, and long
Yeah.
as contraception can lower your AMH.
Yeah, so does IUD,
Right.
but,
Right.
so does IUD, but in absence of those, I mean, given how, is this the granulosa cell dysfunction in these women?
Maybe, ideally, what you would have is a marker of the granulosa cell health. That's really what you want. I mean, that's sort of what you're getting with AMH, but interesting to know. I don't know,
Because
there's a way-
I'm
you
just
could-
wondering.
I mean, what I would predict would be if you can measure their granulosa cell pool, that would probably just give you more information.
Apropos, I saw yesterday a patient who has been for 20 years on birth control pill and has no AMH and has been told for 15 years that she is in menopause.
So who's carrying the ball for this now? Are you still doing this, or?
Well, I'd like to take this work forward in my own lab. I mean, I'm still very interested in the topic, and again, I see these patients all the time in my own clinical practice. I will say I need fertility colleagues who want to provide granulosa cells. I'm not doing the IVF cycles. So,
You must have somebody out there.
so there's
There's some very good people.
there's people who are interested in the question. But I do think that part of it actually is more of how does this get translated actually clinically?
Right.
Yeah.
And again, the way that I see taking this forward clinically is to repopulate ovaries with granulosa cells that are steroidogenic, not the, ideally, you would want to focus probably at first on women who are more interested in the endocrine effects rather than the fertility effects,
Yes.
right? Just from a safety point of view.
So,
I can ask something, because we're hearing more and more often the concept of injecting stem cells into ovaries. A lot of IVF clinics, maybe not in this country, but outside this country are doing it. Whenever we're using stem cells, there's always a danger of malignancy.
Absolutely.
The ovary is probably one of the organs with the highest risk of malignancy, if I had to guess.
Yes. I can tell you what happens if these experiments don't go well.
Okay, that's exactly the question.
Yes, so what happens if you don't sort the cells well?
Yeah.
What happens if you keep the cells in culture a little bit too long and they start to polarize in the wrong direction? You inject them into the ovaries of the mice. The mice develop malignancies, terrible malignancies very quickly. So there's definitely a safety issue there. Now, what I will say, I think the idea, this isn't just for ovaries. I mean, we see stem cell therapies being purported for every condition under the sun. I think indiscriminately injecting stem cells and assuming that they're going to magically repopulate whatever you want to repopulate is probably fanciful. It might occasionally happen, by chance. I think the key is if you can identify a population that is differentiated in the direction that you want and use that, you have to have someone selecting out the stem cells. So I think the idea of injecting either just embryonic stem cells and expecting them to magically fix a problem, or
Even mesenchymal
or even iPSCs that haven't been. So I would worry, and we see this. I mean, I saw that huge tumor on the thigh of the mouse. If you take unsorted induced pluripotent stem cells and you inject them into an organ, they will form tumors, I mean, by definition. So I think that it has to be a sorted
How do
cell.
you get this past an RFD?
Well, I think you could show, again, I think if you focus on the steroidogenic cell population, I think that would be key. You would have to have a mechanism of monitoring the ovaries very closely. I think, again, that's why I would prefer to focus on women who were, say, in their late 30s, early 40s with ovarian insufficiency and wanting to not have to take 10, 15 years of hormone replacement therapy. Look at that. I mean, your safety mechanism there is the fact that, you know, if they start to develop a tumor in the ovary, then you can take it out. You know, the good news is that probably is going to be a germ cell tumor, not an epithelial tumor, at least. But, you know, that would be the safety concern.
Have you ever, sorry,
Go ahead.
have you ever tried to make parallelisms or maybe, pull some data comparing this technique or like this potential technique with the actual ovarian tissue transplants that many times are done after chemo as well, and sometimes are done actually fairly fast, they don't always wait 10 or 15 years to this transplant, sometimes are done 2 years after chemo, and of course we see most of the times the good endocrine restoration in most of the cases, at least for 2 or 3 years usually. Sometimes for fairly more than that, and always every time we see that the AMH goes up and that they are actually start ovulating, we always assume that it's from the oocytes that we transplant that in the tissue. But I was thinking now maybe also the granulosa cells that you transplanted and that actually grafted on the cortex of the ovary also helped other eggs that were there as well, right? We always just assume that those are the eggs that we are putting back, but maybe maybe there's more than that. I was just thinking all the time about that.
Yeah, that's a good point. It could be it's the granulosa cells you're transplanting. It would be if you had a way of labeling the tissue that you put back in to see which oocytes you ended up afterwards.
Yeah.
Again, if you had a sort of a donor scenario, you could probably do that.
Yeah,
So you
of
can
course,
actually-
mostly it's an autologous always, no? And I was just thinking if retrospectively there will be any way to actually test that, like with all the data that we have just now retrospectively, but I don't think that would be possible. Because how do you differentiate that, But like I just have this hypothesis now, it's very plausible, no?
It's absolutely possible, yeah. So I don't know. It also, one thing that's, I guess, intriguing about this particular approach as opposed to cryopreserved tissue is that theoretically this is not a limited supply. You could keep
Yeah.
You might have to reinfuse them every couple of years. It's possible, but you could also just thaw them out and put them back in. Whereas the ovarian tissue, it lasts for 2 to 3 years typically, and then depletes itself. Or the idea that if you could find a way to safely keep these in culture, it might be a way to not even have to have an autologous infusion.
Yeah, I mean, one of the main issues though with the transplant, right, is like we know that like 60% of the tissue that you're transplanting, it's gonna die pretty quickly within the first 48 hours because of ischemia. So I don't know if, because you were thinking about the clinical application, right, of these stem cells, how would you graft them actually?
So we just inject them directly into the ovarian stroma.
Similar to what we do with like the PRP, similar situation, like a little gel that you would inject literally in the stroma? Yeah, interesting.
I mean, you could inject it either laparoscopically or transvaginally, depending on how you exist.
Yeah.
Yeah, the only problem is if it's an ovary, it's just hard to get to if it's a tiny ovary.
David, isn't the company that you are an advisor, aren't they commercially exploring that?
Comida?
What's the name of the company in New York?
Comida. It's one of their product ideas. Right now, they're mainly working on commercializing IPM, in vitro maturation of oocytes, using the stem cell lines that came from George's lab, not these. So there are lines, and they're very similar. Their eLife paper took a very similar kind of course that you all did in terms of making iPS cells and then sorting them for different properties, including steroidogenesis, including expression of germ cell markers, blah, blah, blah. And so their main product line right now is they want to do low stimulation, low or no stimulation, collect cumulus complexes, and then they get co-cultured with the stem cell lines that do happen to be active steroidogenically. And the idea is they're producing factors that are going to improve cytoplasmic quality in the end. But they have other ideas for kind of using, moving these iPS cell lines down different lineages for not just ovarian restoration, but other female reproductive tract tissues.
Okay,
Thank you so much.
thank you.
Yeah,
Thank
yeah.
you very much.
No questions.
Okay, and we're even on time.